Dear friends, this discussion is for blastocyst transfer. I got lot of question through Mr. Deven. He gets lot of questions, and sometimes it happens in my clinic also, that even though when we give transfer good blastocysts which are looking very good, morphologically very good, with good trophoectoderm, with good inner cell mass, with a grade of 5A, with a very easy embryo transfer, with a very soft catheter under ultrasound guide, sometimes these pregnancies they don't occur. I am having Miss Ishani with me. She just completed her postgraduate master's degree from Leeds University, UK. She had her previous biotechnology field also. I am asking her what is her opinion regarding why sometimes these blastocysts they don't implant.
So before we go into how they don't implant, we need to understand that the embryo is graded mostly based on morphology. Even today, even if there is more technology advancement and more techniques to grade embryos, most embryo grading, even if it's by human intervention or by time lapse technology, is only morphology based. So by morphology, we cannot say that the embryo is genetically competent or incompetent, because both of them grow in a similar speed, in a similar manner. So both of them look equally good. But what is going on in the genetic level, we cannot tell by just looking at it. So if we do a morphology grading and a scoring and then implant the good looking embryos in the uterus, then sometimes the pregnancy does not occur. That means
So, what is your view regarding the people? They tell you that they did a PGS for the aneuploidy screening, yes, and they did a euploid embryo transfer even though it didn't plant. Yes, yes. What is your message regarding PGS? Why PGS is not 100% foolproof to these embryos?
So when a practitioner is doing PGS, we are taking a few cells from trophoectoderm, and
so I explain the what is trophoectoderm. This is the blastocyst, and blastocyst has two parts. One is inner cell mass. This makes the baby, and this is trophoectoderm. There is a cell layer over here. So what you are doing a trophoectoderm biopsy.
So in the three compartments, the only group of cells that we can extract safely with the least invasiveness is the trophoectoderm.
So you are taking these cells.
We are taking these cells. So we can do many clinics do a biopsy on day 3 where there are 8 to 10 blastomeres.
But this cleavage
stage biopsy is invasive, and you can only extract one, maximum 2 cells to get any results. And also there is a phenomenon called mosaicism which happens in
What is mosaicism?
Mosaicism, to define in simple terms, means there are two genetic components in a single group of cells, in a single tissue. So some of the cells will be euploid, some of the cells will be aneuploid. So if we are taking blastomeres in cleavage stage, then there is a possibility that the cell that we are extracting, because we can only do single cell biopsy in this case, the cell that we are extracting can be euploid or can be aneuploid, because the mosaicism, the genetic variation within this entire embryo among the blastocysts, is different in this stage, and there is 90% difference in these group of cells.
But the thing is, if you do a cleavage stage biopsy, the chances of implantation
reduces. It is invasive. You are taking cells from the proper embryo which will form. It hasn't differentiated into this. It hasn't differentiated into trophoectoderm and inner cell mass. So you are essentially taking out embryos cells which will form the future embryo.
That is future baby.
Future baby, yes, which will eventually make a fetus and then be born as a baby. So this is a more invasive procedure, and it has been proven that cleavage stage biopsy they reduce the competence, the developmental potential of the embryo. So it's better, the gold standard now is doing trophoectoderm biopsy, because you get more cells and the degree of mosaicism, the degree of difference between genetics in the cells decreases.
Between the inner cell mass and the
The
trophectoderm.
trophectoderm, yes.
But it doesn't make sure that if whatever you are seeing in trophectoderm is 100% or
100%.
is in the which is going to make a fetus.
Fetus, yes,
So
because
there
they
might
are
be a discordance.
maybe a discordance. We can't tell for sure that is there a correction mechanism? Is there similarity between two? There is a correction mechanism because people find out that the cells that are aneuploid in this stage become euploid in this stage.
The cells which are chromosomally abnormal in
In
cleavage,
stage become normal
become normal in the
in trophectoderm.
blastocyst
Yes,
stage
in
in
blastocyst.
trophectoderm by correction method.
Yes, we don't know what that correction method is yet. That is the newest research that is happening in embryology.
Okay.
So why does these good-looking blastocysts, even after PGS, do not implant? That is because there can be some discrepancy that happened when we took out these cells from biopsy, and the cells that we get more number of cells than cleavage stage.
Okay.
We can because this is not forming the baby. This grows rapidly, and you can take out 6 to 10 cells in trophectoderm biopsy. But if there is more mosaicism,
this is a discrepancy between this and this.
No. Discrepancy between the cells here and between the cells here and between these two groups of cells.
Okay, this is called mosaicism.
Mosaicism, yes. Any tissue or a group of cells which has two genetic variants in the same group is mosaicism. So whatever cells we are extracting, we are finding euploidy in them. Means normal chromosomes.
In trophectoderm
biopsy. Suppose I did trophectoderm biopsy, I did a PGS, and I found that there is 46 chromosomes healthy without any structural aberrations or any abnormality. But actually what's happening is there is more number of aneuploidy which is present in the embryo, which I could not see because the cells that I got
were
euploid.
Can't you touch these cells?
No. This is making the baby. So if you take cells out of this, if you put a needle through the inner cell mass, then you're affecting the embryo, which is very, very developed at this stage. So if we take out cells from this lump of cells, there are definitely chances of structural abnormalities in the fetus.
So it is not possible to do an inner cell mass biopsy.
No, never recommended to do a inner cell mass.
But you have tried blastosynthesis also.
Yes, we have done. I studied so blastosynthesis basically is taking out the fluid which is between. There is a fluid which is pumped out when the inner cell mass and trophectoderm is formed. So when this is formed, there is liquid inside the cavity. So we take out the liquid from the cavity.
So I am making the things very short. Why this is not getting implanted in my clinic is not only the good-looking blastocysts are genetically competent. There are other confounding factors. The other confounding factors may be, which I read, and this is what she is telling is actually by evidence, and I am putting this book. It is a collection of lot of research papers, and when you see, there are number of confounding factors which happens.
Whichever I said was the embryological side.
Yes.
So this is what's happening meanwhile in the lab while the patient is getting prepped for implantation.
Okay.
So this is something that's happening in my dish. So this is the embryological side, and now you will tell the gynecological side.
The gynecological side is that if this embryo is, even though PGS is done, that is PGTA aneuploidy test, and it is found that it is 46XX or 46XY euploid. So you had a question that I transferred euploid, we do PGS and we will do euploid transfer, but even though there is no pregnancy,
Yes.
there are multiple confounding factors, whether the mother is accepting this embryo or not, whether this endometrium is in the implantation window or not, There are multiple factors that is immunologically acting over here. So even though if you see this, that when a transferring single euploid embryo, live birth rates are 40% to 60%. That means there are 50 to 40% they are rejected because they look good but genetically abnormal. And even though they implant, 15% of them miscarriage. That means the total pregnancy going on, if you make 15% out of this, the maximum it goes to 45%. So even though if you transfer a good looking blastocyst with PGTA, the chances of getting pregnancy, if other confounding factors is other factors we nullify, it becomes 45%. After this transfer, there are multiple factors from the mother, that is activation of coagulation system, implantation window, there is a immunological rejection. All these factors, they make the things more complicated. As we reach a top of the mountain, we need lot of things with us. We need a good boot, we need good equipments, we need good clothing, we need oxygen. Even if one factor is deficient, like if I don't have a good boot with me, I cannot climb. So there are multiple confounding factors in, even though there is a good blastocyst, good looking. I think even by time lapse technology, they are not 100% sure that
sure.
Even though
With time lapse technology, the only thing that is done these days, and mostly time lapse is better used in a research condition where you're studying whole embryos, you're even studying the inner cell mass, blastocele, and the profectoderm. That case, a time lapse machine is useful. But in a clinical situation, the only thing time lapse does is it doesn't let you take out the embryos again and again outside. It stays in there for 5 days. So in my experience, when I compared time lapse with the drop culture that normally we do, the hatching and the blastocyst formation was roughly the same.
That is very important criteria, that time lapse and with the micro drop technique which we are using in our lab is almost the same, and the ultimate end result is getting a good morphologically graded best blastocyst. So this, thank you very much Ishani, and this is the discussion which I put, that why a good blastocyst looking good, time lapsely looking good, even though it is transferred well, not getting pregnancy. The reason we explained. Thanks a lot for patient listening.